Today I'm going to be talking about a method we're developing for the analysis of single-cell RNA-seq data called STREGA-NONA, or single-cell transcriptomics, reveal extended gene set associations in networks optimized with a genetic algorithm. So some analysis of large-scale data sets uses co-expression networks, which are data-driven networks based on large-scale data sets such as transcriptomics or proteomics, and edges represent associations between genes or proteins. And these can be made for a specific context like viral infection. You can then use network metrics for mechanistic predictions or to predict the impact of perturbations to information flow through the network. Knowledge-driven gene sets are based on mining primary literature for interactions, which is a very time-consuming process. So some of these aren't updated very often, and these databases contain gene sets with general cellular processes. And not all of these databases account for network topology, but these functionally related gene sets are often used for the interpretation of large-scale data sets. For example, a large-scale bulk RNA-seq data set from peripheral blood mononuclear cells from infants with RSV was clustered based on gene expression. Each of these numbers is the size of the cluster. And then we used overrepresentation analysis to see how these clusters were enriched in knowledge-driven gene sets. So a few problems with this type of analysis is we still get very dense networks, and the overrepresentation analysis isn't always specific. Like each cluster maps to multiple gene sets, and multiple gene sets map to different clusters. To improve on this, we're trying to optimize cluster and gene set specific subnetworks with a genetic algorithm. So we're taking single-cell RNA-seq data, clustering the cells based on expression, calculating a mutual information matrix to generate our co-expression networks. From there, we do random walks starting from our genes of interest to sample the network around these genes and get a starting population. Then we use an objective function to optimize our genetic algorithm and get optimized extended gene set networks, which we can then compare between experimental groups for differential network analysis. So a genetic algorithm takes a population of networks and does mutations and crossovers. So this can be the deletion or insertion of nodes or edges, or crossovers where sections of the network are swapped. And this sampling gives us a random selection of nodes and edges around our genes of interest. And these are optimized using prior biological information. Specifically, we're looking at single-cell RNA-seq data from peripheral blood mononuclear cells from people living with HIV. For this study, we have 8 subjects, 4 with atherosclerosis and 4 without atherosclerosis. And here you can see the clustering of these cells. And this plot here shows the different clusters with the number of cells from each experimental group within that cluster. So it varies between clusters. And we actually excluded the smallest cluster as it didn't have enough cells for analysis. So our cluster-specific co-expression networks were made by first filtering out genes with counts less than 1 and greater than 90% of cells. And then we calculate a mutual information matrix for all gene pairs and filtered those gene pairs based on their mutual information if it was greater than the mean plus 2 standard deviations. So again, we have the clusters in order of size, with the largest ones having actually the smallest percentage of genes kept after filtering, and the largest ones keeping the most genes, which you can see here in this plot where we have the number of cells and genes after mutual information filtering, where the smallest clusters have the highest number of genes kept, which could be due to the fact that these are likely more similar as the clusters aren't as spread out. To actually get our starting population, we're doing random walks along the co-expression networks, starting from our genes of interest. We looked at a few different lengths of random walks, because if we have long walks, we get networks with more overlap between different gene sets. And if we do walks that are too short, we don't get a large enough connected component for analysis. We ended up using 2 times the median shortest path between genes of interest in the co-expression network as the length of our random walks. because this gives us small enough networks so we don't get a ton of overlap between gene sets, but small enough that we get a large connected component. So specifically, when we run this GA, we're looking at immunology-related gene sets from MSIG-DB, and we're including gene sets with at least 15 genes in the co-expression network. We start with an initial population size of 50 and run for 20 generations. At each generation, we make 100 child networks using mutations and crossovers, and then take the top 50 networks from the parent and child population based on the objective function to move on to the next generation. Our final population is then put into one network where the edges are weighted based on the number of times an edge appears in the final population. And then the shortest paths between each gene pair are calculated to get our final weighted networks. Our objective function incorporates prior biological knowledge to get the most biologically meaningful network. So we're maximizing a score between 0 and 1. This is an example of the overall objective function for different gene sets across 20 generations. So the parts of our gene set, the parts of our objective function include the grouping of genes predicted to be the target of the same transcription factor, a comparison to the co-expression network from the other experimental group, since that helps with differential network analysis later. We look at the number of genes from the starting gene set that are still found in the network, because if we delete all of the starting genes, that's not a gene set specific network anymore. We compare to a network made only from cells closest to the centroid of the cluster, since these are likely to be the most similar in gene expression. And we look at the eigenvector centrality of genes from our starting gene sets to make sure they still have a high impact on the information flow through the network. Before optimization, we have varying number of nodes and very dense networks, as you can see here from the number of edges in our starting networks. After optimization, though, regardless of the starting network size, we get very similar number of nodes and much smaller number of edges. So we're definitely optimizing the number of edges in our networks so they're more interpretable. Looking at the eigenvector centrality as a network metric for our data set, we looked at 914 gene sets across 15 clusters. So these had at least 15 genes in that cluster. The black on this plot represents gene sets that are not found in that cluster. We found that most of our T cells clustered together, as well as our B cells, and that in our T cell clusters, we actually found a lot of T cell related gene sets that have at least 15 genes in our networks and a higher eigenvector centrality. Looking at one specific cluster, we have preliminary results from our naive T cell cluster. And here we're looking at the difference in eigenvector centrality between the AS positive and AS negative group. So anything that's 0 is the same eigenvector centrality for both groups. And we found genes related to T cell activation and proliferation that were differentially connected between the two groups. As an example of what our optimized networks actually look like, this is the same naive T cell cluster and a cytokine stimulated T cell gene set, where blue nodes represent genes found only in one group. Pink are found in both groups, and if they have a purple outline, those are our starting gene set genes. So we can actually see how different the connectivity of these genes are between different groups. In conclusion, we've included prior biological knowledge in data-driven single-cell co-expression networks to help with mechanistic predictions. Some analysis of large transcriptomic data sets, such as data-driven clustering and overrepresentation analysis, are not very specific. Random walks sample networks around the gene of interest while making a population of unique individuals. And eigenvector centrality and other network metrics show the impact of perturbation of genes on networks. In the future, we'd like to validate with simulated data. Our actual data shows that the normalized abundance of all genes is lower than the normalized abundance of only genes found in MSIG-DB gene sets. So we can introduce a signal as our gene sets for simulated data. We would also like to include gene sets from KEGG in our analysis and test our algorithm on another single-cell RNA-seq data set of PBMCs from infants with respiratory syncytial virus. With that, I'd like to thank my PI, lab members, my committee, and the people who actually collected and analyzed the HIV atherosclerosis data and our funding. Thank you.